Hi, I came to image my samples.
Hi, I'm Gopi Shah. I'm the project manager for advanced mesoscopy applications. For imaging large biological samples up to a size of 1 millimeter, we use a multi-view light sheet microscope. So in light sheet microscopy, you illuminate the sample with a sheet of light in one plane, and the sample moves through the sheet of laser. A camera is continuously recording images, producing a three-dimensional stack of images of the sample. Now, this particular system has 4 lenses, 2 of them for illumination and 2 for detection, so that you can acquire 2 opposing views in a sample simultaneously. If the sample is too large, we rotate the sample to acquire 2 more views. That way, we end up with 4 or more views to generate a 3-dimensional reconstruction of the entire sample. So for imaging living samples, such as developing embryos, spheroids, organoids, we require a very robust environmental control. So a system such as this one regulates temperature, humidity, pH, gas content, and so on. So while imaging multiple samples in this system, the system acquires images from below, illuminates the samples from the side, and it can image dozens of samples in parallel. It could either be many samples of the same kind, or it could be samples being imaged under many different environmental conditions.
I'm Montse Coll, Mesoscopic Imaging Specialist.
In the Mesoscopic Imaging Facility, I handle the clear sample related projects. Here in the wet lab, we make the samples transparent to improve the propagation of light through the tissue. And what we do first is to embed the samples inside a block of agarose to be able to handle them properly in an optical projection tomography or light sheet system. After embedding the samples, we proceed with the clearing protocol. What most of the tissue clearing methods do is to replace the lipid and water content of the tissue by a higher refractive index solution that matches the refractive index of the remaining elements of the tissue. This way, we reduce light scattering and we increase the penetration depth for imaging.
Hi, I'm Jim Swoger, and I'm the head of the Mesoscopic Imaging Facility. So this is an optical projection tomography machine, OPT. It works a bit differently from the light sheet. Instead of having the thin sheet of light, we illuminate the whole sample and we rotate it instead of scanning it linearly. So the sample attaches onto this arm here, it rotates. We can lower it into this immersion fluid, and then we're imaging it in transmission with the microscope here. It also works in fluorescence. So what happens is we collect a bunch of images, rotating the sample and taking pictures from different angles, like an X-ray CT machine. And then that raw data can be reconstructed to give you the volumetric data, in this case, the interior of the juvenile octopus.
Hi, my name is Adrias Cubat, and I'm an imaging technology specialist at MEF. At MEF, we not only use commercial microscopes, but we also develop our own techniques adapted to the requirements of our users. For example, here we have a light sheet microscope that was designed to image large cleared samples. We can image organoids or mouse brains of up to several centimeters in size. The microscope offers dual-sided illumination with 2 opposing objectives in order to generate a large and uniform field of view. It also has 4 different detection objectives that you can choose from and image samples of different sizes.
Hi, Jim. I came to pick up the data. Do you know if it's ready?
Yeah, we've got 2 terabytes here for you.
Okay. Wow, that's a lot. Can you help me analyzing it?
Sure, we'll help you process it. It's not the end of the world.